Hi everybody, today I want to start with the carbohydrate or saccharides are the most abundant organic molecule in the nature, or we can say one of the most abundant molecule also in the biological system. And the general formula is the CnH2OM. It can show the relation between carbon and the water. The minimum number of the carbon is 3 or bigger than 3, and with this classification we can start with the carbon number 3. It is a trios. O's meaning is sugar. 3 is the number of carbon. We have two types, aldehyde and the ketone. Aldehyde, glyceraldehyde. Okay. It is a chiral molecule. We can draw here in the both shape. I draw the mirror image also. It is a L-glyceraldehyde. And if you see the mirror image, it is diglyceraldehyde. Both of them has a chiral center. They are non-tumor each other. Mirror image. But in the ketone, the first molecule is the dihydroxy acetone, which is not chiral. I can draw here. No chiral or achiral. In the sugar which are aldehyde, we can say aldose. And if the ketones, we can say ketose. Okay, we can add the number of the carbon also here. For example, we can say glyceraldehyde, it is a aldo trios. And the hydroxyacetone is the keto trios. It is the first group of the carbohydrate with 3 carbon. We will see in the next lectures these 2 compounds we can find in the biological system also, which is very important. If we carbon number increase, for example, if we have a more carbon, 4 carbon, it is a tetrose. Example for tetrose, we can write erythrose. If the number of carbon is 5, we can say pentose. The example for this one, it is ribose or deoxyribose is we can find in the DNA and RNA. If we have a 6 carbon, it is a hexose. We can find in the glucose example or fructose. And also we have a 7 carbon in the biochemistry. We can see it. It is a heptose. For example, we can say pseudoheptose. You will see in the shunt. As you see here, these classification is related to the number of the carbon. And with the carbon, usually the number of the water also is the same as the number of the carbon. Okay. In some books they write C H2O n times. Carbohydrate is very important because they have many functional in the body as energy source, also in the membrane of cells, in cell membrane for recognition and connection between the cell. They are the backbone or the base of the anabolism for the production of the other compound also. For example, after glycolysis, we can see how they affect in the formation of the other compound. The second classification of the carbohydrate, we can start with the number of the unit of the carbohydrate. The first unit of the carbohydrate is the monosaccharide or monosaccharide. Then we can classify as a unit of carbohydrate. For example, if we have a 1 unit, it is a monosaccharide. Monosaccharide, from the 5 carbon, we can have a 2 shape, linear or acyclic and cyclic. It is very important, usually from the ribose and deoxyribose and hexose, we have this classification of the cyclic and the linear in the biochemistry. If we have a 2 unit, it is a disaccharide. Then 2 unit of the monosaccharide bind each other and remove the water and formation of the disaccharide. For example, lactose is formed from the galactose, which bind in the beta form from 1 to 4, bind to the glucose. For example, I can draw in the cyclic form, and in the carbohydrate in the biochemistry we usually use the D form, D-galactose and D-glucose. I use the cyclic, it is a galactose, it is the epimer of the glucose. As you see here, it is a galactose. If you see the beta, here is the beta up, connection to the carbon number 4 of the glucose. What is the difference between glucose and galactose? It is they are epimer in the carbon number 4. As you see here, in the galactose, OH in the number carbon 4 in the up, but in the glucose, it must be in the low. And when they bind each other, they lose of water and the formation of disaccharide. Then two monosaccharide with the remove of the one water, they form glycosidic bond. Then we can say it is a glycosidic bond, which is the reason of the binding of two monosaccharide with each other. And if we have a more than 2 unit until 9 unit, or we can say oligosaccharide. Carbon 3 unit, of monosaccharide to the 9 or 10. This is the oligo. We will see, for example, we can say example in the dextrine or other relations. We can also speak about this. Now I want to speak about relation of the monosaccharide. For example, if we have a 6 carbon, we can have a 4 isomer: glucose, galactose, mannose, and fructose. Then I can write 6 carbon and 6 carbon isomers. I can write glucose, galactose, mannose, and fructose. Fructose is the ketose, but these 3 are aldose. The relation between the glucose and the galactose, we can say C4 epimer, and the relation between glucose and mannose, they are C2 epimer. I will explain now in the example, and you can see how is the relation. I drew glucose with red, okay, it is a glucose, 1 aldehyde, carbon number 1, and the carbon number 2, OH right, number 3, OH left, carbon number 4, again OH right, carbon number 5, OH right, the carbon number 6, CH2OH, which is not chiral. First, I show the chiral centers. They are chiral centers, so I put a star here. 4 carbon chiral center we can see in the glucose. It is a glucose. It has a 4 chiral center. 2 power 4, it shows the number of the isomers, optical isomers. So it has a 16 optical isomer. It is 2 power n shows the number of optical isomer. Now I want to show epimer. It is a glucose. Now, for example, if I copy this molecule again, as you see here, if I change carbon number 2 OH group and H group, I choose the other color, green, for example. It is OH here and H here. As you see here, the carbon number 2, in carbon number 2, which is optical active, we change the position, then it will be C2 epimer, and it is a mannose. And again, if I change again color to yellow, or here the brown, and if I found the carbon number 4, it is a carbon number 4 compared to the glucose, and if I change the position of the OH and H in the here, you can see will be galactose. All of them are D. As you see, the last chiral center or carbon number 5, if the OH group in the right side, in the last chiral center, it will be D, or left side will be L. Then it is a D glucose, it is a D mannose, and it is a D galactose. But fructose, it is a ketose. For the drawing of the fructose, first carbon must be CH2OH, because carbon minimum position of the ketone, it is in the carbon number 2. I drew the ketone here, and I continue exactly the same as the glucose. If you see the glucose, the same situation, carbon OH number 3, carbon 4 OH in the right, carbon 5 in the OH is again in the right, and in the last one, CH2OH. As you see here, it is a ketose, and the number of the chiral center is 1 less than is glucose. It has the 3 chiral center, then 2 power n is equal to 2 power 3. It is 8 optical isomers it has. Then relation between fructose and aldose or hexose is the structural isomer or constitutional isomer, because they have a different functional group. The first group is the aldehyde, and the second group is ketose. Now, if we can make a mirror image. For example, if we have a glucose here, I copy this molecule. For example, I use galactose. If you see here, if I make a copy of galactose, I'm sorry, I copied the glucose, but no problem, I correct it. If I change I do the mirror image of the galactose, then I change the position of the OH and OH in the chiral center. The first one, OH, H, H, OH, H, OH, OH, and H. It is a L-galactose. Okay, it's good situation. As you see here, OH in the left side, it shows it is a L. And full structure of the molecule is in the mirror image with the glucose. Then we can say enantiomers are a special type of the isomer, which we can find in the pair structure with the mirror image of each other. And the most important, the molecule must have a chiral center. Without chiral center, we cannot do anything for the molecule, and the mirror image hasn't any meaning. And usually for the conversion of these two, we use enzymes which belong to the isomerase family. It's very important. And the other things, what I show you, for example, in the galactose structure, if you see the relation between these two, I draw again here. OH, OH, OH. This molecule is the cyclic galactose. If you see here, it has a hemiacetal. No. In the carbon here, number 1, here in the cyclic, if OH is in the up, it is a beta. If OH is in down, it will be alpha. Then if I draw the like this structure, I copy it. And I change the position of the, okay. This structure we call it is a pyranose. It is very important, the pyranose, because it is from the pyrene structure. You can see in the organic chemistry lectures also. If the OH group in the carbon number 1 in the cyclic form is up, is beta. If the down is alpha. Okay? As you see here, I drew galactose. Then I can see some information about it I will show you. If the CH2OH outside of the cyclic, it will be shown it is a D. If it is opposite, we change the position with this hydrogen. It will be L. Okay? Then both of them are D. In the galactose, as you see here, OH in number carbon 4 is the epimer of glucose. This carbon, OH, it shows it is galactose. Then, if I draw the same molecule here, but just I change here the position of the hydrogen and OH, then will be beta D glucose. But here is the beta D galactose. Okay, for example, the last one, it is a galactose. First I change it to the glucose. Carbon number 2, I change 4, excuse me. And now if I change carbon number 2, it will be, again, here shows a D. Carbon number 2, OH inside is the opposite of glucose, will be mannose, because if the OH is down is alpha. D alpha mannose. Okay, this alpha shows this the cyclic. When we have a alpha or beta, it shows the molecule is the cyclic. But if we don't have it, this alpha in the linear, but we can say also the special name, we can add also, we can say D alpha pyranose. Okay, we can use also this name. It shows it is a pyrone because we have a two shape of structure in the sugar. If we have this structure, it is a pyranose. If we have this structure, it has a furanose. Usually for the fructose, we have a furanose, and with glucose family, we have a pyranose. Also with ribose, we have a furanose, excuse me. And the relation between these two monosaccharides in the cyclic form of the glycosidic bond. Glycosidic bond usually produced between the carbon number 1 and carbon number 4 or 1 from the second sugar. And the position it must be shows is the relation between the glycosidic bond can be alpha or both beta. For example, if we have an alpha 1,4, it means the first monosaccharide in the carbon number 1 bind to the carbon number 4, but the first monosaccharide is alpha. Always, when we draw this situation, it shows when it is alpha, it's oxygen here. For example, if the beta oxygen is up, it is very important. Here you see, it is alpha. For example, bind for the other cyclic. For example, iodopyranose, if you see here, we can say alpha 1, 4, because the carbon number 1 is here. 1, 2, 3, 4, the second one is here. It is a 4. And 1 is here. And position is alpha because it goes down. And it's very important for here. And how the linkage can be happen and how can be classified. The last part of the glycosidic, which is normal glycosidic, is so C glycosidic is between the carbon and the carbon. But sometime we bind the sugar to the protein or amino acid. For example, I have a glucose, as you remember, it is a alpha glucose, or can be monosaccharide, or we can continue with the polysaccharide or disaccharide, no problem. But the first monosaccharide you need can bind for example, amino acid you need is, for example, asparagine, which is bind in the protein. It is a protein molecule. Then we can use a protein. It is a sugar. And the unit is the starting material is asparagine, ASN. As you see here, we say N-glycosidic bond. N-glycosidic bond formation. What is this? As you see here, for example, the relation can be happen one hydrogen from here and one OH from here, remove of the water. And these two molecules bind here each other. Then I can make a copy. And here I make a paste. As you see here, this OH group cancel, this hydrogen group cancel, and they bind each other. As you see here, it is an N-glycosidic bond, which shows how is the binding of the sugar unit with the protein. We can see in the cell membrane this type of structure, very important. Or sometimes we bind protein. For example, I again make a copy of the whole process. Okay, for example, the first amino acid in the protein I use serine. As you know, serine in the R group is the CH2OH. It is a serine. And if we want to bind together, the glycosidic bond is O-glycosidic. And the remove of the water can happen between these two molecules. In this situation, this part will be changed. or I can make this difference. And this OH will bind to CH2 has one oxygen bind for this carbon, and it's called O-glycosidic. Again, it is a protein bind to the sugar with the amino acid serine, which is very important, the relation and study. In the first lecture, I spoke about the mainly relation about the biochemistry and organic chemistry in this chapter. In the next chapter, I will try to teach digestion of carbohydrate, which it is one of the source of the energy in our body, and mainly we use it. I hope it can be good for you, and see you in the next lecture.